Dear students, Assalamu alaikum. Towards the later part of last lecture, we studied the physiological anatomy of the urinary bladder. The objectives of today's lecture are to learn about bladder filling, micturition waves, micturition reflex, and abnormalities of micturition. Transport of urine from the kidneys through the ureters into the bladder. Urine flowing from the collecting ducts into the renal calyces stretches the calyces and initiates peristaltic contractions. These spread to the renal pelvis and then downward along the length of the ureter, forcing the urine from the renal pelvis towards the bladder. These peristaltic contractions are increased by parasympathetic stimulation and decreased by sympathetic stimulation. The ureters course obliquely for several centimeters through the bladder wall. The normal tone of the detrusor muscle in the bladder wall tends to compress the ureter and therefore preventing black backflow of urine from the bladder. Filling of the bladder. When there is no urine in the bladder, the intravesicular pressure is about 0 centimeters of water. 0 centimeters of water. And by the time 30 to 50 milliliters of urine have collected, the pressure rises to 5 to 10 centimeters of water. Additional urine, about 200 to 300 milliliters, can collect with only a few centimeters of water rise in pressure. This constant level of pressure is caused by intrinsic tone of the bladder wall itself, because the bladder muscle has the property of plasticity. Before proceeding further, it is important to understand this concept of plasticity. When smooth muscle is stretched, the tension initially produced is not maintained. Is not maintained. Therefore, despite an increase in volume, the bladder wall is capable of maintaining intravesical pressure at only 5 to 10 centimeters of water. The relationship of bladder volume and the intravesical pressure can be explained by the law of Laplace. The law of Laplace states that the pressure in a spherical viscous is equal to twice the wall tension divided by the radius. In the case of the bladder, the tension increases as the organ fills, but so does the radius also. Therefore, the pressure increase is slight until the organ is relatively full. A plot of intravesical pressure against the volume of fluid in the bladder is called a cystometrogram. The cystometrogram shows an initial slight rise in pressure, then a flat segment, and then a sudden sharp rise in pressure. Beyond 300 to 400 milliliters, as you can see in the opposite figure, collection of more urine in the bladder causes the pressure to rise rapidly. Now, superimposed on the tonic pressure changes are periodic acute increases in pressure shown by the dashed spikes. These pressure peaks are called micturition waves. These peaks may rise only a few centimeters of water or may rise to more than 100 centimeter of water, and they are caused by the micturition reflex. In the cystogram shown in the previous slides, we can see that as the bladder slowly fills, micturition contractions begin to appear. These contractions are the result of a stretch reflex, which is initiated by sensory stretch receptors in the urinary bladder wall, and especially by the receptors in the posterior urethra. When this area begins to fill with urine at the higher bladder pressures, sensory signals from the bladder stretch receptors are conducted to the sacral segments of the cord through the pelvic nerves, then reflexively back to the bladder through the parasympathetic motor nerve fibers. As the bladder continues to fill, the micturition reflexes become more frequent, causing greater contractions of the detrusor muscle. Once a micturition reflex begins, it is self-regenerative in nature. That is, initial contraction of the bladder activates the stretch receptors to cause an increase in sensory impulses, causing a further increase in reflex contraction of the bladder. And this cycle is repeated again and again until the bladder has reached a strong degree of contraction. After the bladder has reached a certain strong degree of contraction, the self-regenerative reflex begins to fatigue. The regenerative cycle of the micturition reflex ceases, permitting the bladder to relax, and the intravesical pressure dropping back to the basal level. Therefore, the micturition reflex is a single complete cycle of progressive and rapid increase of pressure, a period of sustained pressure and return of the pressure to the basal tone of the urinary bladder. As the bladder becomes more and more filled, micturition reflexes occur more and more often and more powerfully. Once the micturition reflex becomes powerful enough, it causes another reflex which passes through the pudendal nerves to the external sphincter, to inhibit it, that is, to relax it. Now, if in the brain this inhibition is more than the voluntary constrictor signals to the external sphincter, urination will occur. If not, that is, the voluntary constrictor signals are more than the inhibition, urination will not occur until the bladder fills still further and the micturition reflex becomes more powerful. At a volume of about 600 milliliters in the urinary bladder, the rhythmic contractions become very strong. Urination can only be suppressed by a continuous conscious effort. And this conscious control has a certain limit. That is, when 700 to 800 milliliters of urine accumulates in the bladder, voluntary control exerted by the higher brain centers fails. So the detrusor goes into a continuous contraction and micturition takes place. Now let me talk about the facilitation and inhibition of micturition by the higher brain centers. Now, although the micturition reflex is an autonomic spinal cord reflex, but it can be controlled by centers in the brain. These centers include strong facilitative and inhibitory centers in the brainstem and inhibitory centers in the cerebral cortex. Now, as I said earlier, although the micturition reflex is the basic cause of micturition, but the higher centers normally exert final control of micturition as follows. Final control. The higher centers keep the micturition reflex partially inhibited, except when the micturition is desired. They can prevent micturition even if the micturition reflex occurs by tonic contraction of the external bladder sphincter until a convenient time presents itself. So, when it is a suitable time to urinate, the cortical centers can facilitate the sacral micturition centers to help initiate a micturition reflex and at the same time inhibit the external urinary sphincter so that urination can occur. Okay, now this figure depicts the micturition reflex and its control by the higher centers. On your left hand, the figure shows the stretch receptor, sensory efferents, and the motor impulses coming via parasympathetic nerves to the urinary bladder. And on your right hand, the figure shows the motor impulses from higher brain centers stimulating relaxation of internal and external sphincters, hence signifying the role of higher centers in the control of micturition. Okay, now let's study some of the abnormalities of micturition. Atonic bladder. Now, if micturition reflex contraction cannot occur, if the sensory nerve fibers from the bladder to the spinal cord are destroyed. So, a person loses bladder control despite intact efferent fibers from the cord to the bladder or normal neurogenic connections within the brain. So, if the sensory fibers are gone, the reflex is no more there. So, what happens? Instead of emptying periodically, the bladder fills to capacity and overflows a few drops at a time through the urethra. This is called overflow incontinence. The causes of atonic bladder include crush injury, like in road traffic accidents, to the sacral region of the spinal cord, or diseases causing damage to the dorsal root nerve fibers that enter the spinal cord, like syphilis. Now, this condition is called tabes dorsalis, and the resulting bladder condition is called tabetic bladder. The second disorder of micturition is the automatic bladder. Automatic bladder is caused by spinal cord damage above, I repeat, above the sacral region. So the sacral cord segments are still intact, and typical micturition reflexes can still occur, but no longer controlled by the brain. So initially, after the damage to the cord has occurred, the micturition reflexes, like other reflexes, are suppressed because of the sudden loss of facilitative impulses from the brainstem and cerebrum. However, if the bladder is emptied periodically by catheterization to prevent bladder injury caused by overstretching of the bladder, the excitability of the micturition reflex gradually increases, and periodic but unannounced bladder emptying occurs. Another abnormality of micturition is the neurogenic bladder, which results in frequent and relatively uncontrolled micturition. This condition results from partial damage in the spinal cord or the brain stem that interrupts most of the inhibitory signals. So there is absence of inhibitory signals from the brain. Therefore, facilitative impulses passing continually down the cord keep the sacral centers excitable. So excitable that even a small quantity of urine elicits an uncontrollable micturition reflex, leading to frequent urination. Therefore, a neurogenic bladder is uninhibited. It is caused by lack of inhibitory signals from the brain, and it results in frequent and relatively uncontrolled micturition. Another abnormality of micturition is dysuria. Dysuria means difficulty or pain associated with voiding or passing of urine. The causes may be cystitis, which is the infection of the urinary bladder, urethritis, infection of the urethra, or it may be due to a stone in the urinary bladder, causing the infection, stasis and infection. Vesicoureteral reflux. Now, in certain individuals, the contraction of the bladder during micturition does not always lead to complete occlusion of the ureter, and therefore some of the urine in the bladder is propelled backward into the ureter, a condition called vesicoureteral reflux. Now, this reflux, if persistent, can cause enlargement of the ureters because of holding more than urine, and an increase in the backward pressure in the renal calyces and structures of the renal medulla, and therefore causing damage to these regions.